Hi, we're here today to talk to you about engineering an emergency ventilator for COVID-19. I'm Dr Joseph Sherwood, and I'm going to introduce the talk to you, and then we'll hear from some of my colleagues on various aspects of the project. So how all this started is around the issue of ventilation and COVID-19. So you see here in this picture a gentleman who is intubated. This means he has a tube down his throat, and that intubation is connected to a ventilator system that is doing the job of breathing for the patient because he's no longer able to breathe for himself. Jakob, our clinician on the project, was working on the front line in the Royal Brompton Hospital and talking to colleagues in Italy where the outbreak was severe at the time. And so around mid-March, he came to the Department of Bioengineering asking for help. So why did he come to the Department of Bioengineering? Well, we have to start with answering the question what bioengineering is. So if we look at some of the research topics ongoing in the Department of Bioengineering at Imperial, they're quite varied. So we have biomechanics and mechanobiology, imaging such as MRI scans, CT scans, etc. We have molecular and cellular bioengineering, understanding how mechanics actually interacts with molecules on the cellular level. We design medical devices, we do computational and numerical modeling, neurotechnology and robotics, and regenerative medicine and biomaterials. So there's a lot of overlaps with various other sciences, but the thing that really holds it all together is that bioengineering is the application of engineering principles to biological and medical problem solving. And so today we're going to talk to you about a specific problem and how our team came about solving it. So the problem was ventilator shortage. So there are many ventilators available from a huge number of different manufacturers. This picture here shows a Draeger ventilator, which is one of the leading companies based in Germany. And all of these ventilators have complicated designs with specialist components, which is an appropriate approach to take for a market that requires a slow overturn of ventilators. So it doesn't matter to have the complexity, and you can spend a long time perfecting how the device functions. The problem is when COVID-19 came along, the global need for ventilators increased incredibly rapidly. And it was estimated that there's a need of 1 million new ventilators, and the scale of production in the world is just not possible to produce that number of ventilators. So there was a need for low-cost, easy-to-manufacture ventilators for COVID-19 patients, and that's the problem that we addressed. This is a little video of our final solution, and then what we're going to do is tell you in this talk how we got from that problem to the solution. This is actually a computer-aided design model of the final system that's been rendered to make a nice video. Back around to the front. Okay, so here's an overview of the talk. We're going to start off with Jakob, who's going to tell us about the clinical need for the ventilators. Then I will move on and give it a little bit of background of the physics of breathing and how a machine can take that job on for us. Then Michael will teach us about how to actually build JAMVENT and the processes that went into that. Then Jennifer will talk us about how to build a model of the human lung for testing purposes. And finally, we'll give you a very brief history of the first 5 weeks of the JAMVENT project with Jakob.
Okay, so thanks for that, Joseph. So without further ado, my name's I am a clinician. So just a little bit about me. So I'm actually also an Imperial Medicine grad. So I did my undergraduate medical degree at Imperial, graduating in 2012. What do I do now? Well, I'm a senior anaesthetic registrar, and for those of you who don't know what registrar means, effectively that's the stage just before I become a consultant. And I'm also a bit of a frustrated engineer, so I'm always looking at the kind of technical manuals, all the devices we use. And one of the things that attracts me to anaesthetics in particular is the kind of working on basic working on problems using basic kind of principles, which kind of marries quite well with engineering. I've been kind of frontline on the COVID pandemic, so here's me in my full kind of PPE get up. This was actually with one of the team's background, so this was me doing a video call from one of our hot zones at the Royal Brompton Hospital where I work out to one of our clinicians kind of doing the paperwork outside. I'm also a research fellow at the college, so kind of in my spare time, actually, I work on kind of machine learning type techniques to improve risk prediction in emergency surgery, and that's one of the areas of research I do. I've got a couple of other side projects, too many in fact. And what's my role in JAMVENT? Well, I'm the clinical lead. As Joseph mentioned, kind of I was hearing these stories from colleagues who had friends in Italy and kind of just how dire the situation was in sort of Bergamo, and contacted the Department of Bioengineering, kind of kicked off the project, and then have been kind of doing the clinical input primarily, although I'm trying to get my hands into the engineering as much as possible where I've been able to. And so basic things. So what do lungs do? We've got two of them. They take up most of your thorax. So as you can imagine, they're pretty important. And so primarily they are a massive area for diffusion of gases. And so when you breathe in air, which contains 21% oxygen. You want that oxygen to go into your bloodstream. And very importantly, you also want to get the carbon dioxide that your body produces when it metabolizes that oxygen to kind of do basic bodily functions and get that out of the way. But lungs are a lot cooler than that. Actually, they do lots of really important stuff. So actually, drug metabolism is a really important function of the lung. So actually, when a drug goes into the body or any kind of chemical, it's broken down sometimes to inactivate it, sometimes to activate it. And the lung is a really important area for that. And very interestingly, it has a big role to play in the kind of way that we measure blood pressure control in the body. And the mechanism behind that is linked to COVID, and also is potentially one of the reasons we've seen differences in disease severity in different ethnic groups. And so just a quick warning now, do look away if you're a little bit squeamish, because I'm going to show you what a pair of lungs looks like. So this is a pair of lungs kind of being inflated. These are pig lungs, which are actually slightly larger than humans, but basically the same sort of size. And you can see they're very sort of soft jelly-like material, kind of going up and down. And you can see that they're really fragile. It's actually really fragile tissue that's really, really easily damaged. And so now I'm going to tell you kind of what COVID actually does to the lungs. And so when I talk about COVID, actually what I'm really talking about is the virus. So that's SARS-CoV-2. And so it's a coronavirus, and corona means round, as you expect. So like you have a corona in a solar eclipse. And so it's this round virus with little spiky proteins on the outside, which are imaginatively called spike proteins. These act as keys, effectively, that go into a lock. And so the lock in this case is a receptor, so called ACE2. And that's actually one of those things that I was talking about was involved with blood pressure control. And so what happens is SARS-CoV-2 uses this kind of lock and key type mechanism to enter cells and starts causing damage. And so just in this example, so here at the top is kind of effectively what normal lungs look like. You've got 5 lobes, 2 on the left, 3 on the right. And actually it's made up of these tiny little tree and bud type things, and these are called alveoli. And so normally it's all very homogeneous, lots of little buds which take part, which is where the gas exchange takes place. Now, if you have an infection, as you have down here, as my friend Tom's kind of drawn, effectively what happens is the body tries and fights off that virus. And what happens when it does that is it creates, it releases lots of inflammatory mediators, so lots of kind of chemicals that try and damage the virus, attract other cells to try and help do the immune response. You get fluid kind of coming into the area of kind of infection, which is inflammation. It's almost like, if you think of like a sunburn on the inside. And what happens is all these alveoli get plugged off with kind of mucus and pus. And here you'll see that you'll get basically you get narrowing of the airways. So as the airways become inflamed and you get this kind of inflammation, the airways narrow, and that's kind of the reasons you get lung problems with sort of lung dynamics. And so what does this look like? So I'm going to turn you into radiologists in about 60 seconds. So what we have here is a normal CT scan. So this is kind of one of those the CAT scans, so the kind of donut you often see people going through in TV shows. In this case, white here, so this is a spine. So white and bone dense things are white. Black is air, so here's an airway here, and you can see it's very black. This is a heart. These are ribs around the outside. You've got lungs. This one is the right one. This one is the left, because actually you're looking up from the feet. And you can see actually that both lungs are very black here. You've got some white bits in there, and actually that's the blood vessels that have a dye in them. And you can see it's all very evenly distributed, big surface area for gas exchange. Now, what happens in severe COVID-19 is you get massive inflammation in areas of changes. So you can see immediately there's in this patient, there's a lot less black areas, a lot more white areas in the lung. This is actually a late stage disease. So actually, if you look here at this yellow arrow, what we have is an area of what's called fibrosis. So actually where the inflammation has progressed to a point where actually what that soft jelly-like substance has now become almost wooden and sort of fibrosed. And here is just some area where actually the kind of that kind of nice spongy lung is contracted down and is kind of pulling the airways open. So it's really not taking effective point in effective use of gas exchange. And so what do we have to do then? So we have to place patients on a ventilator, generally because of that impaired, that there's no longer that massive area for gas exchange, we have to help support them. And what we do is, as an anaesthetist, we'll give them an anaesthetic, or use something called a laryngoscope, which we place into the mouth and kind of pull the jaw forward. And that allows us to look down into the windpipe, the trachea, and place what's called an endotracheal tube from the mouth into the trachea to kind of connect them up to a ventilator. And this is a pretty fundamental principle. So I do it in humans, but actually some other examples are a lion. So here, this vet here isn't even using a laryngoscope. And gorillas also get intubated, in this case for an ultrasound scan of the heart. So it's a cross-species procedure. I'm just going to hand you back over to Joseph now.
You're muted, Joseph. You have to unmute yourself. Sorry.
There we go. Thank you, David. Yes, so I am a Royal Academy of Engineering Research Fellow in the Department of Bioengineering. And that means the Royal Academy of Engineering support me to do research. So I came at this subject through mechanical engineering. Like many people who study mechanical engineering, I originally wanted to build motorbikes. I thought that would be the coolest thing to do, until I discovered that I could study blood, which is a lot cooler. So this is actually a video from my PhD work. This is a plastic vessel that's the size of a hair, and those are human blood cells flowing through it. And one of my research topics at the moment is how mechanical changes to the red blood cells in diabetes affects the flow, and how that might actually play a role in the disease. I also study the flow of fluid in the clear part of the front of the eye and how that regulates intraocular pressure, which is a key component in the disease glaucoma. I'm also lucky enough to teach in the department. So part of the third year course called Biomechanics, I teach biofluid dynamics in that course, and you'll get a little taste of that today. And my role in JAMVENT is the project and the technical lead. So we're going to start with some basic physics. This may be very familiar to a lot of you. Some of you, this may be new, or some of you joining who aren't students or soon to be students, this may be very old for you, and it'll be some recollection. But most of this stuff is actually intuitive, and we deal with pressure all day, every day. So it's just a matter of kind of formalizing that. So if you imagine this dark circle here as a container, and inside it we have some molecules of gas, and they're all bouncing around. And then every now and then you'll get molecules of gas bouncing off the walls of the container, and they apply, when they make a contact with the wall, a force. And if you consider that over the whole area, the pressure is the force divided by the area over which that force is acting. If we add more particles into that same container, we'll get more collisions, and that means we'll get more pressure. If we take the same number of particles, but we make the container bigger, you're then going to get fewer collisions, just because statistically the times when they hit the wall is going to be reduced. And so that means you get a lower pressure. And so this last relationship can be summarized by something called Boyle's law, which states that the pressure multiplied by the volume is a constant. So in this top case, we've got a certain pressure and a certain volume. If we increase the volume, we lower the pressure. And if we decrease the volume, we increase the pressure. So that's a key thing to know about what pressure actually is. Now, pressure is important in this context because it drives flow. So we imagine we've got these two containers here, one with a high pressure and one with a low pressure, and we connect them to each other. Then what you'll get is a flow of these gas particles from the high pressure into the low pressure region. And you get this, if the pressure difference is relatively low, you'll get a relatively low flow rate. And the volume divided by the time is what the flow rate is. So the flow rate tells you how much actual stuff you can move in a given period of time. If we now consider this is a much higher pressure, you're going to get a higher flow rate because the gradient in pressure is larger. And so the flow rate is proportional to the difference in pressure. So controlling the pressure allows you to control the flow rate. But the flow rate also depends on constriction. So if we imagine that same situation, but now with a much narrower corridor here, then you'll get far fewer particles passing through. In the simplest way of looking at it, it's just because there's not as much space for the individual particles to pass in. A way of thinking about this is if all of these little dots running around here are students in one classroom and they have to rush to the other classroom to get there quickly, if the corridor is narrower, it's going to take that whole classroom longer to get across because there's just not space. Additionally, you're going to be bouncing into each other and knocking each other off track. And so it's going to take even longer. And that's actually what turbulence is. It's all the molecules of air bouncing and randomly, chaotically moving around. So the constriction both increases the turbulence and also it's going to increase the frictional forces between the molecules and the wall, which will decrease the flow rate for a given pressure. So Jakob, as a clinician, sees a patient and will know what volume of fluid he wants to give to them in each breath. That's called the tidal volume VT, and how quickly he wants to do it, so in a time. So a constriction of the airways, like you showed us earlier, will mean you're going to have a higher pressure in the lung. So how do we breathe, firstly, under normal conditions? This is a little animation I found which does look surprisingly like an unhappy frog, but that has no relevance to the topic. So what we do here, this green band here is a picture of the diaphragm. And when you want to breathe in, your diaphragm contracts, it pulls down, and that increases the volume of the lung. And at this moment in time, imagine that there's been no change in the number of particles of air in the lung. Now we already know that as the volume increases, with the same number of particles, the pressure is actually going to decrease because of Boyle's law. So now we have a low pressure inside the lung and a high pressure outside the lung, and that's going to drive a flow into the lung, and that is how we inhale. The air will continue to fill the lungs until the pressure inside and outside the lungs is the same, which will be the atmospheric pressure, so the pressure that's around us all the time that exists because of the weight of all the air molecules on top of us. And then to exhale, it's actually a passive process. Even though you can actively exhale, what happens under normal breathing is all those alveoli Jakob showed us earlier, basically made out of a material called elastin, which is similar to a rubber band. So when we breathed in and increased the volume of the lung, those rubber bands get stretched. And then when we breathe out, the rubber bands constrict again, and that forces the fluid back out of the lungs and causes a small positive pressure inside the lungs during that process, but a very small one. So here's a little animation of breathing. So what about a ventilator? So as you can see here, we've got a diagram of normal breathing. So on the left-hand side, we've got the atmosphere, so the kind of reference pressure outside the lungs. And then on the right-hand side, we're showing the pressure inside the lungs. And so we've just seen that you actually create a vacuum inside the lungs, and that draws fluid into the lungs. But we can't do that during ventilation because we can't control the diaphragm. So instead, what we have to do is apply a high pressure from the ventilator and use that to drive fluid into the lungs. But very critically, we're now at a very different range of pressures than the lungs are used to working at. And as Jakob pointed out, the lungs are quite fragile tissue. So it's important to avoid the pressure going too high, which could cause barotrauma. Baro, if you think barometer, and trauma damage. So at the end of each breath, the pressures are equalized in the lung and outside the system, so the ventilator or the air. So during exhalation in a normal patient, you get that slight positive pressure I mentioned, and air flows out from the lungs back to atmospheric pressure. In ventilation, because we have this bad general function of the breathing, what we want to do is do everything we can to make sure that the alveoli stay open. So actually at the end of the breath, rather than going back down to atmospheric pressure, we go to something called PEEP, which is peak expiratory end pressure. And so that's another design feature that's required for a ventilator. And this keeps alveoli open and makes gas exchange more efficient. So those are kind of idealized pictures of what happens, but here are some actual waveforms. So this is the pressure. You can see during inhalation, it goes up, hits about 30 centimeters of water. So that's if you had a column of water that was 30 centimeters high, that's the same amount of pressure in that. And at the end of the breath here, you see the pressure never goes below this set value of PEEP that the clinician would have decided upon. These are the flow rates, so you can see where it flips over here. That's as you go from inhalation, the flow is positive, exhalation, the flow is negative. And then these are the volumes delivered into the lung and then out of the lung. So the lung volume is changing here by about ½ liter in each breath. The clinician sets this parameter VT, which disappeared there, and you can see that we aim to meet that exactly during each breath. So my final slide here will just be a little segue into going from those basic concepts to how you end up coming up with a design. And you need design principles to do that. Now, as I mentioned, engineering is a problem-solving task, but it's actually a trade-off between what design options you have and real-world factors like cost, quality, and time. So we can consider this triangle of cost and time and quality. And in this context, quality would be performance of the machine and different modes of operation, for example, and time would be related to ease of manufacture and hence supply chain options. So existing ventilators are very high quality, they have lots of functionality, but they're slow to build and they're expensive. Now I should note, typically these triangles are thought of in terms of choosing two and letting the other one go, but actually the real practice of that is slightly more complicated. For the simple emergency designs, they're quick and easy to build, but they're relatively low performance. So they will really address the problem of the rapid need, but not always the problems of actually what's needed to help COVID-19 patients. So with JAMVENT, we try to find a middle ground. We get all the quality that we need to help the COVID-19 patients, but also to be able to quickly manufacture at relatively low cost.
Thank you, Joseph. I will take you through how to build a JAMVENT machine. So first off, a little bit about me. I'm Michael Madekurozwa, and I was born in Zambia and spent the first 8 years of my life in Lusaka. I spent the next 7 years in Zimbabwe because I'm Zimbabwean by descent, and then I came to the UK for my A-levels and stayed here ever since. So I did my undergraduate and PhD in bioengineering at Imperial College London and wanted to enter medicine at graduate entry stage, but I liked bioengineering so much I've stayed inside it ever since. Currently, I'm an Imperial College London visiting researcher and a private consultant. I joined the JAMVENT team because I thought it was a fantastic opportunity to apply the skills I have learned as a bioengineer and to learn from a world-class group of engineers and make a positive difference. So I'll go through a little bit of the concept of how the JAMVENT system actually works. So the first step is to allow the patient to exhale. After the patient has exhaled, the JAMVENT system prepares a breath at the desired air to oxygen ratio and volume. Next, the system delivers the breath to the patient. And then this cycle repeats. So the patient is allowed to exhale, a breath is prepared, and then the breath is delivered to the patient. And this is what ventilation is. So I'll go through a schematic of the system so that you can get a better appreciation of how it actually works. So first of all, we've got a reservoir. This is where the air to oxygen ratio, the air to oxygen mixture is prepared and then at the correct tidal volume before being delivered to the patient. We need air and oxygen. So an air and oxygen supply are connected to the system as shown in the diagram over here and here. And then we've got pneumatic valves to help control the flow of gas. So here are the pneumatic valves, B, A, C, and D. So these are just on-off solenoids. In high-end ventilators, these are typically actuated solenoids that change their diaphragm. So they're not just on and off; they actually change the size of their diaphragm. So this means that they're specialist components, a little bit difficult to source, which puts a bottleneck into the supply of ventilators. We opted for on-off solenoids, which makes the design a little bit more difficult to make, but it makes it more readily available. So inside the schematic, whenever I show a solenoid in this formation, that's the on position. It's allowing air through it to go into the reservoir. And in this position, that's the off position. It's not allowing air to go through it. There's a pressure sensor inside the system over here, which is used to calculate the pressure in the reservoir, and this is used to titrate a volume for the patient. So this is used to calculate volume via Boyle's law, which Joseph spoke about earlier. The pressure sensor, there's another pressure sensor inside the system, and that's down over here, which measures the pressure inside the lung. So this is for clinical use because it's good for the clinician to be able to see what's actually going on inside the lung. And it's also as a safety mechanism. So if the pressure inside the lung goes high, the system will detect it and act accordingly in order to decrease the pressure. There's an oxygen sensor inside the system down over here, which measures the actual oxygen to air concentration that's being delivered to the patient. And there are also pressure release valves inside the system. So the pressure release valves are over here and over here. These help make sure that the system pressure doesn't go too high as a safety mechanism. So ventilators usually have a lot of safety mechanisms inside of them because as Dr. Mathiszig-Lee mentioned earlier, there's a lot of safety that has to go into these devices as the lungs are very, very delicate organs. So ventilation. So the first stage of ventilation is exhalation. The patient is over here. Valve D opens, allowing the patient to breathe out. So air goes through valve D and out of the system. Note that valve C is closed, so air cannot go around this way. After exhalation occurs, a breath is prepared for the patient to breathe in. So the air and oxygen lines open, so valve A and valve B open, allowing air and oxygen to flow into the reservoir. Once enough air and oxygen have flown into the reservoir to deliver one breath to the patient, the air and oxygen lines close, as shown in this figure over here. So the air and oxygen lines are closed, and the single breath is delivered to the patient by the opening of valve C, straight down here to the patient. And then this cycle repeats, and this is how ventilation occurs. So here's a schematic of what the system looks like, and I've been showing you a schematic all along, but it's not actually what the system looks like. So here's a 3D rendering of the system. So you can see that the pneumatic solenoids actually are like this, and the manifold is like this, and the pressure sensor is like this, and so forth and so on. And here's the system with the lid on it, just to show you what it looks like when it's complete. So this 3D CADing mechanisms are used in order to help us design the system, know which components we're going to actually have inside the entire system and how they'll all come together. And here's an overview of the assembly of the system. So typically, ventilators are made out of specially custom-built pneumatics, and these are difficult to source and also difficult to make. We built the entire JAMVENT system out of off-the-shelf push connectors and threaded connectors, which means a lot of the connectors are very easily replaced and also easier to source. Now, all along I've been talking about the pneumatics of the ventilator. There's also the electronics of it, and the electronics we have put at the bottom of the ventilator. This means it's easy for someone to work on the pneumatics without ever disturbing the electronics, and vice versa. Easy to someone to work on the electronics without ever disturbing the pneumatics. In actuality, here's a picture of what the electronics looks like. So we've got a bunch of modules inside of here that are off-the-shelf modules and wires connecting them together. So all that's needed to actually assemble the JAMVENT system is a pair of wire strippers and a screwdriver. And here's a schematic of what the system looks like with all the connectors between the modules. So I've talked about the hardware, and this hardware needs to be run by something. And it's run by software that's written on a GUI, which is run off a computer. And here's a schematic of what the computer software looks like, which Dr. Frattolin will go into in the speech that comes just after me. This GUI is written inside of LabVIEW, and here's a view of what the code actually looks like. So this is an overview of the code. I'm not going to go into detail about what exactly it is, but it's composed of several different blocks, several different functions. And this is just an overview of how those sub-functions are connected together and how it's written in a sort of modular function. So what skills actually go into making a JAMVENT ventilator? There's electronics, computer-aided design, an appreciation of the physiology, an understanding of pneumatics, control engineering, computer programming, and data analysis. All skills that we have honed as bioengineers. I'll now pass you on to Dr. Frattolin, who will talk about mimicking a human lung.
All right, perfect. Thank you, Michael. So my name is Jennifer Frattolin. So I was born and raised in Canada, and I did my PhD in mechanical engineering from McGill University in Montreal. And I was a person who loved medicine, but didn't want to become a clinician. So I decided to go and do research in bioengineering. So currently I am a research associate here at the college in the Department of Bioengineering, and my research is focused at the interface between the lymphatic and the immune systems. And so I'm involved in JAMVENT as part of the technical team. And the reason why I joined was because I have a keen interest in medical devices. And I also wanted to be able to use the skills I have developed as a bioengineer to be able to help others during this pandemic. So why do we need to now test this ventilator that we have designed and built? So it's a multifaceted kind of answer here. So first of all, we need to evaluate the performance of JAMVENT at regular intervals throughout the design process. So we need to create a design, build it, then we need to test it, and then we need to evaluate the results of that testing to make an informed decision on whether or not the design is sufficient. And likely this is going to be an iterative process until a finalized design is actually realized. And once we have that final design, then what we can do is we can present it to regulatory bodies to demonstrate its safety and efficacy, and hopefully achieve regulatory approval so that we could then use it in a clinical setting. So how do we actually test a ventilator then? Well, particularly during the design process, it's really not logical to utilize human testing, and there's lots of ethical concerns, obviously, regarding human testing. So instead, we need to fabricate a lab bench system to test our ventilator, combining our knowledge both of lung physiology and the principles of engineering in order to be able to simulate the behavior of a lung. So this kind of brings us to the question of, well, what do we actually need to mimic? So when we talk about the lungs, there are two main principal components. So you have the lung compliance, and then you also have the airway resistance. So when we talk about lung compliance, we're talking about a measure of the lung's ability to stretch and expand to be able to hold air that we inhale. And when we talk about airway resistance, we're talking about the resistance or the opposition to airflow in our respiratory tract due to friction. So, but what happens to our baseline lung compliance and the airway resistance during respiratory distress, such as with COVID-19? So does the lung compliance and our airway resistance change from its healthy state? And of course, the answer is yes. So these values are dynamic and they will change depending on the severity of the disease. So when we talk about lung compliance, as a lung disease progresses, typically, like Jakob said, we see an increase in fibrosis of the lungs, so an increase in that hard kind of wooden-like tissue, and this is going to decrease the compliance of our lung. And when we talk about airway resistance, with respect to lung disease, we remember Jakob talked about the inflammatory response of our airways. Often that is accompanied with some swelling. So we see a narrowing of our airway, which will lead to an increase in the airway resistance. So we now know that we need to build sort of a test system that has variable lung compliance and airway resistance in order to take into account varying states of disease severity. But how do we actually do that? So with respect to simulating lung compliance, we wanted to use really basic engineering principles. So to do so, we utilize the concept of a leaf spring. So if you can imagine, we have a plastic sheet and that is fixed at both ends. And as we apply a pressure to the bottom side of that sheet, it is forced to bend. And how much that plate will bend depends on the stiffness of our material. And by changing the stiffness of the material, we can actually alter the overall compliance of our lung. So the other thing we wanted to really focus on was using easily sourced materials. So for example, you can see in the video, we use a typical kind of hydration bag that you would use for hiking to act as our lung. And we utilized easily to obtain Perspex sheets that could be layered in order to act as our compliance, but that could also be variable. And we used a sort of aluminum hinge on either side in order to anchor the Perspex sheets, completing our leaf spring. Finally, we really wanted a design that was simple to construct and easy to replicate. So by using the concept of the leaf spring, it was quite easy to build and it didn't require a lot of complex materials. And as you have seen within this video, it's actually relatively easy to assemble and disassemble. Now, what about the airway? How do we replicate that airway resistance? So in order to increase airway resistance, we need to increase the friction that the air is going to be exposed to. So to do so, what we do is we use a combination of wire and fabric gauze. And by placing this in the flow path, this changes the size of our airway into a series of very tiny little airways, which increases our overall surface area that the air is actually exposed to, and thereby increases our friction. And what we can do is we can actually layer these wire and fabric gauzes in order to modify, so to increase or decrease our resistance. And we can use a 3D printed holder that we've made to actually hold everything in place. Then we use some tubing connectors to hold this and allows this to actually connect it overall into our test system, which you can see here. So on the left-hand side, we have our actual ventilator, which is then connected with our resistance. In the middle, you can see with the arrow. And then on the right-hand side, we actually have our test lung, which represents our compliance. Okay, so now we are equipped to be able to test our ventilator, but how? So as Michael said, we've developed a software interface that allows us to control and manipulate the various different ventilator modes and parameters. So on the left-hand side here, you can see our user manual, where a clinician like Jakob is able to change the input parameters of our ventilator, such as our respiratory rate or our tidal volume. And then on the right-hand side of our panel is the calculated patient outputs, which are updated with every cycle. And if I start the video, you can actually see that occurring. And in the middle of the interface, you can see our pressure and flow rate waveforms with respect to time. And finally, on the right-hand side of the screen, you can actually see one of our test lungs inflating and deflating. So the scenario that's kind of depicted here is one of a severely diseased lung. So we have a very low compliance lung with a high resistance in the airflow path. So, and this is really evident by the high peak inspiratory pressures that you can see on the right-hand side of the screen. So it looks like PIP, and that are observed during inhalation. But when you actually look at the test lung, you can actually see that the lung itself is inflating very minimally. And because of this, we have modified our ventilator settings accordingly. So for example, we are now delivering a much smaller tidal volume. So we're delivering a less volume of air, but more frequently, so at a faster rate.
Okay, so finally, I'm just going to take you through the kind of brief history of how this happened and how quickly all these things come together when you've got a really good, high-quality interdisciplinary team working together. Just to take you through the timeline. So this is our first prototype, so the Mark 1A, which is basically a very basic system just to prove that we could kind of get some sort of gas flow. I sent the initial email to the Department of Bioengineering on the 15th of March. And so we had this first initial prototype working 4 days later. 3 days after that, we kind of managed to progress a little bit. And actually at this point, we're now being able to control volumes through that kind of same system. And here we're using actually just a simple party balloon to act as the reservoir, which is flexible. And I think this was a really elegant solution because it reduced the amount of dead space in the system, but unfortunately it's just not quite robust enough to use kind of long term. Mark 2 came on March 27th, so 2 weeks later. So this is one of the systems that I am kind of most fond of because I kind of came after night shift to help assemble this, was doing a lot of the wiring. But actually you can see here within 2 weeks, we fundamentally got the same system that we have in our latest prototypes with the valves, the reservoir and the electronics, but in this case, just in a sort of breadboard type system. The Mark III, within a month of me sending a completely speculative email about potentially building one of these, now looks like a real ventilator, standalone with a portable PC, and now this actually looks like a medical device. And now, a day after that, a week after that, we got a suction system working. So actually, we managed to maintain pressure during suctioning of the patient airways, which other ventilators were really struggling with. And 2 days after that, we had spontaneous mode working. So that's the system responds to and then, sorry, can someone just mute their mic, please? And so this is me going through the system there. So just showing that we've got the Mark IV hooked up to a head. Sorry, hang on. I'm just going to mute everyone.
I've muted them.
Okay, great. I'm just going to replay that. So sorry, this is just me demoing the system. So this is where we are, where we've got the Mark IV system hooked up to a tablet. We've got some ventilator tubing connected to an endotracheal tube. And here we've got a decapitated head that I stole from actually the set of the new Batman film, so look out for that in a few months' time. And we're hooked up to our test lung. And here I'm just demonstrating that actually now with a touchscreen interface, we could adjust the parameters as you saw before. And so just to close up, we've got some thank yous.
Thanks, Jakob. Yeah, I just wanted to finalise by saying thank you to everyone who's been involved in this project. There's been a huge number of people who've donated their time for free, many of whom are mentioned on this screen. So in addition to Jennifer, Michael and Jakob, who you've heard from today, Professor James Moore has been involved from the very beginning. He actually teaches medical device entrepreneurship in the department, and that's a great course. It's produced lots of successful companies so far, and hopefully JAMVENT will be another one of those. Daniel Watson, who's also in Professor Moore's lab, who did the animations for this and helped Jen with the test lung. And then Axel and Willie, who helped us with the software and modeling the system. There's a large number of students below, as well as Satpal, who did some machining for us. Also, people from throughout the college, the Icon team, Enterprise. A friend of mine, Nina French from Hello Hackenbacker, helped us with all the branding. So we've got the nice color scheme, etc. And then finally, we also want to thank the people who helped fund this research. So we were generously awarded some money from the Imperial COVID Fund. That started off our processes with the manufacturers we're working with. The Royal Academy of Engineering and the Wellcome Trust are funding some of us who are actually working on this project. And the Department of Bioengineering have been amazing in their support, and we really want to thank them and everyone else at the college who we weren't able to mention explicitly. And with that, thank you very much, and we'll happily take questions.
I'm going to give an applause on behalf of the audience. Thanks to the four of you for this incredibly interesting talk. I guess it's fair to say that it's nothing short of impressive what you've achieved in that short period of time. And I think also a wonderful illustration of the wide range of skills and knowledge that really is needed and that you acquire if you become a bioengineer. So I think for many students in the audience, that will be very nice to see how they really learn and will then be able to combine a broad range of engineering skills to build a machine like this. But I don't want to take up more time than I already have, and instead would like to open the floor for questions from the audience. So if people have questions, if they would like to raise their hand with the little hand button that you see in the taskbar, if that's the right word, of Teams, then I try and call you up without butchering your names too badly. And if you remember that you please address your questions directly to one of the four speakers that we just had, and that will be great. So if people have questions, please raise your hand, and I try and fairly pick representative of order, although the list of people in the audience is long, so I might miss people. So please raise your hand if you have questions. And I'm sure there are many. Just saying that. There's one. Emily, you can start, please.
Sorry, that's a talk question. I'm going to talk. Can you hear me?
Yes.
Fantastic. This is a question for Jennifer. I don't know if you're in a position to comment on this, but I was wondering if you could say, give an overall judgment at this stage on modelling of lung function. And generally, if we are moving into another, well, not another paradigm, but has that been greatly increased during COVID? Or is it too early to say?
Can you hear me?
I can hear you.
Excellent. Oh, sorry, I was saying unmuting on Teams. So I'll give a good stab at this, and then I'll probably open up to the rest of the team for their opinion as well. So I think the answer would probably be yes. I mean, COVID has certainly brought a lot of exposure, I think, to respiratory disease and the respiratory system in and of itself. And so as a result, I think, perhaps not an immediate reaction of the pandemic, but I think, later on, there will be more fundamental research in, I think, the overall respiratory system. But immediately, I mean, even in our project, we have done a lot of modeling ourselves of the actual respiratory system. So we had someone, one of our colleagues, Willie, who is completely in charge of actually modeling the overall system of the lung and the respiratory system for us to be able to design the ventilator accurately. So I think, yes, I think absolutely modeling of the lung is a vital point in this kind of pandemic. Does anyone else have anything to add?
Yeah, I guess just to add my point in from the clinical point of view, and I guess it slightly depends on what you mean by modeling in terms of whether you mean kind of physical modeling or whether actually it's about kind of like understanding the kind of pathophysiology of disease. I think one of the things that COVID has shown is actually a lot of these kind of ARDS, so acute respiratory distress syndrome diseases like, for example, seasonal influenza, which we've primarily thought of as respiratory diseases, are actually probably vascular diseases to a large extent. And so, as I was saying, actually a lot of the damage is kind of inflammation of the lung tissue itself. Actually, there's a significant burden of the disease that is vascular in origin. So actually lots of little blood clots that are clotting off the blood. So even if you have an area of lung that's well ventilated, there's no blood flow getting it to allow it to kind of take that that kind of gas out. And actually at the Royal Brompton, where I've been working, we kind of pioneered the routine use of something called dual energy CT scans, which allowed us to look at the kind of microvascular disease state of the lungs in these patients. And actually we saw a huge, a really interesting burden of kind of microvascular disease, which is going to be really important in terms of the long-term comorbidities. So, COVID is terrible is actually a fascinating condition. I think there's going to be, PhDs upon PhDs about kind of lung physiology going forward on this.
That's very helpful. Thank you. That's exactly what I wanted to hear. Thank you very much.
Thanks. I'm going to take a question from the meeting chat by Mark Atkinson, and I guess this would be one for Joseph and or Michael, so you can fight between the two of you who wants to take this. And it's a question of how you can make your device more applicable to low and middle income countries. And the concrete question is whether you will be producing a compile control software that would run in a device like a Raspberry Pi to make this work.
I'll take the first part of that, and then I'll let Michael talk about the electronics side of it. So for low and middle income countries, there are obviously several challenges there. One is supplies of oxygen. So we originally designed this to operate with the hospital supply line oxygen, but we did some testing at low pressures. So oxygen concentrators, which are fairly widely available, can generate a bar of pressure, which is about a quarter of the hospital supply lines. But we've configured the system so that it can actually operate fully functionally at these lower pressures. We haven't built our own oxygen concentrators, so we would need to source those separately, but that would work in tandem with that as well. The other big advantage for the LMIC, I don't want to use the word market, but the people who need it in those countries, is the ease of maintenance of this system. The only moving parts of those 4 valves, they're designed so they can be very easily taken out and replaced. And that means that you won't end up with big warehouses full of complicated equipment without the technical expertise to maintain it. Michael?
So on the side of the electronics, the entire JAMVENT system, just components-wise, costs under £2,000. So it's quite cheap. And then the electronics are very simple inside the device. It's made of nothing more than a couple of power supplies, a couple of programmable logic controllers, and then assembling it is very easy because all you need is just a screwdriver and a pair of pliers, essentially.
Yeah, thanks, Mark. I hope that answered your question. If not, just add another comment. Sorry, Joseph, did you want to add something? I just saw.
Yeah, there was a question as well about
Raspberry
code to run off Raspberry Pi. Yeah, so we actually throughout this project have had a team of engineers and clinicians over in Boston who have been actually working on developing code for a Raspberry Pi that would run the JAMVENT system. also, one of our manufacturers has been developing code for an Arduino type system, but a medically approved Arduino type system. So there are teams running those programs. We've provided a quite detailed publication of how it all operates. Our intention being that people could take it up and build it for the hardware that they can source.
Okay, thank you. There's another question in the chat by Roshan, and I guess this might be one for Jakob and Jennifer, so you can maybe share that between the two of you. The question is, for patients who have COVID-19 and need some form of help with their breathing, would the JAMVENT ventilator be better than the CPAP device made by UCL and Mercedes? Or maybe you can talk about what the differences are between CPAP devices and the JAMVENT.
Yeah, so it's an interesting. It's basically effectively a CPAP device requires that the patient be awake, and effectively CPAP means continuous pressure. So you just apply continuous pressure to the patient, generally via a face mask. So effectively, you can use non-invasive ventilation for COVID-19, but there's certainly the early evidence, and generally the big bulk of the evidence is a lot of patients with severe disease. Even if they initially do well on CPAP, inevitably needs invasive ventilation anyway, just from a physiological point of view. And so that's one of the reasons why I pitched this as a kind of invasive ventilator. And they're complementary technologies, not necessarily, it's what either or. And there's some significant issues with the CPAP device that UCL and Mercedes made. It's a really elegant system from a kind of engineering perspective in that it generates pressure using kind of Venturi effects and that kind of stuff. But it uses a vast amount of oxygen. And in a pandemic situation, actually, you're limited by the amount oxygen. Hospitals, the supplies of oxygen for hospitals comes from a big tank of liquid oxygen, and actually that's limited partly by the diameter of the pipes in this hospital, but also the kind of limits of evaporation from that liquid oxygen to a gas, and how quickly you can heat it to a temperature that's not, you don't want to give minus 160 Celsius gas to patients. So actually, the UCL systems use 70 liters a minute. Potentially for sick patients. Ours is really, really highly efficient. Actually, a friend of mine who works at UCL sent me a video of all the oxygen low pressure warnings going off in the hospital when they only had a handful of patients on those systems. So they're complementary, and they kind of treat different stages of the disease. Yeah, I hope that answers your question.
Cool, thanks. There's sort of a related question, as far as I understand anyway, from one of our undergrads, Arush. Where does the exhaled air go? Is there a filter to prevent contaminated breath from entering the ambient air?
Yeah, so I can sort of tackle with that, but effectively there are in fact two filters. So there's a filter at the patient end, and there's also a filter at the kind of exhalation port of the ventilator. And so actually these are really high efficient viral filters, and so they scavenge all the kind of viral particles.
Okay, great. I can't see any raised hands, but there's more questions in the chat, so I'm just going to give you another one from Mark. And the question is there an appetite to have two expiratory control valves to enable more complex methods of ventilation in the future, like airway pressure, APRV, airway pressure release
ventilation?
Yeah, okay, that's what it means. There you go. Jennifer or Joseph, that's probably one for the two of you, maybe.
I mean, I am not completely clued up on all the various different ICU ventilation modes. This is one that I've discussed with Jakob. The general idea here is that if you're going to start adding proportional valves in, you're going to need flow sensors, because one of the elegant parts of the JAMVENT design is you avoid the flow sensors because you can use the flow pressure properties of the valve. Now, if you have a valve that changes its flow pressure properties, then you can no longer use that to measure flow. So in addition to having to add those proportional valves, you would then also have to add flow sensors. At that point, you're really heading back towards the standard designs, which we're deliberately trying not to encroach upon. Jakob could maybe answer if there's any benefit to doing something along those lines.
Yeah, so I think we talked about APRV, and there's potentially the chance that we could probably already do it anyway. The way we do our suction mode is actually relatively similar to kind of APRV. We could adjust the code to do it. I think in terms of what we thought about in terms of, particularly with this rapid cycle time, in terms of trying to concentrate our efforts where it would be most effective, sort of my final decision was actually, if someone needs APRV, they're going to be on one of the kind of existing ICU ventilators that an ICU might have. And so rather than go for that initially, concentrate on the fundamental kind of modes that we needed.
There's a question by S.E. Greenwald. Please go ahead.
Hi, yeah, very interesting talk, extremely well presented and very enjoyable. My question's a general one, which is, can you tell us something about the regulatory barriers that you're presumably dealing with and may well have dealt with already?
Yeah, so that's been a fun one. So the MHRA process really didn't work very well for us. They kind of made their decisions within a week about who they were going to run with. And so despite us publishing all the evidence that we can meet all of the requirements, so that's including the spontaneous and the suction mode, that did, as we showed earlier, take us 5 weeks from the beginning of the process. So by that point, the MHRA had actually decided they were not going to take any other devices forward. Some of the ones they did, in fact, almost all of the ones they did, they then dropped because they couldn't do those functions. So we've turned our eye towards the US. We're in the process. In fact, on Tuesday, we resubmitted another 300 pages or something of documentation to the FDA in response to the first 100-odd pages we'd sent through. So we're trying to get the emergency use authorization from the FDA, which we hope will come through in a few weeks. That won't officially approve us for use outside the US, but actually in practice, the World Health Organization have said they will recognize FDA EUA approval as proof of safety for these devices for emergency systems.
Thank you.
Thanks, Professor Greenwald, for the question. I see that Jakob has just answered another question on the chat, which currently leaves me out of questions. So I invite the audience again to raise their hands or add questions to the text.
I guess I'll just, if anyone isn't reading the chat, I'll just quickly kind of say what I said, which is Mark Atkinson asked if we were considering a turbine version to kind of get over the hurdle of compressed gas. And again, one of the things is turbines, much like proportional control valves, are highly specialized in individuals. So you've got supply chain problems, and you can't just switch out one turbine for another. It also needs a flow sensor partnered, kind of coupled with it. So that's the reason we kind of stayed away from that concept. As Joseph mentioned, actually, we can operate off a bar of pressure if needed, which is what an oxygen concentrator pulls out. So we've kind of said if you're going to do ICU level care, you need some kind of oxygen supply, and we can run off that even if it's from an oxygen concentrator.
Thanks, Jakob. I think there's another question in the chat. From Shamus Sim. The question is, are there any more plans to improve the current design of the ventilator?
I do have one improvement that I would like to make, which is the exhalation pathway. Actually, the original design was not to use a solenoid valve, but to use a pinch valve. So you basically have a disposable piece of tubing and the solenoid valve acts externally on it and compresses it to close the valve and then can open. Much as we've tuned the flow characteristics of the valves, if we correctly tune the flow characteristics of that tubing, we can then just dispose of that piece of tubing after each patient and replace it. And then you don't need that extra filter downstream, and then you reduce the amount of consumables that you would use. So that's the main next advance I would like to make to the system. But again, sourcing pinch valves that are large enough to actually allow that kind of amount of airflow through has turned out to be very difficult to do indeed. So that's why we haven't done it that way so far.
I guess the other thing as well is, as we've mentioned with some of the funding, we've got a new control system in development from one of the manufacturers we've been working with. So that should hopefully reduce the need to have an external computer, which would be a big step forward in terms of robustness.
Arush, go ahead.
Hi, yes, I just had a quick question on the mixing part in the reservoir. I was just wondering, like, how do you ensure that the oxygen and air is mixed completely before sending it to the patient? Because I think, like, wouldn't that be sort of a threshold time before you can sort of send the air to the patient where it would be optimally mixed? Because then the sort of if you sent it too quickly, then it wouldn't be mixed effectively.
So this is partly to do with how fast the air moves into the reservoir. The air moves very quickly into the reservoir, and then the oxygen moves very quickly into the reservoir as well. And that speed just increases that mixing process. There is a little bit of a lag when you change oxygen to air ratios. It takes a bit of time before that reservoir is fully emptied, and then the new concentration is delivered. But that's typically a couple of seconds before we get there.
Okay, thank you.
And just to know, we do monitor the oxygen concentration that is being delivered to the patient, so we know what it is over time as well.
There's a question of one of your colleagues, Sam Al, who first of all pays you your compliments. Says it's very exciting work, and he wants to know whether there's any way to test O2 or CO2 gas exchange capabilities ex vivo.
Would Sam like to explain further what he means by that question?
Yes, and maybe you can join us and speak to us and say
Sure,
that.
yeah, very happy to talk. I mean, this is very interesting work, and I think this is excellent, and I hope all the best for you and all the patients with COVID. I'm just wondering how people do these test the ability of these systems to actually transport oxygen and CO2 to patients before actually going into a patient. So I mean, I guess you're able to show, you're transporting O2 and air into the patient system, the ex vivo patient model. But is there any way to determine, how well it's transporting CO2 out of that model and exchanging gas before you go into patients?
I mean, the only thing I can think of is, I mean, maybe I'm not sure in terms of like modeling the actual kind of diffusion. I know there's systems you can get, which I think they're called like calorimeters or something like that, where effectively they're a candle, a very fancy candle, which burns the oxygen and produces CO2. And that would at least allow you to kind of model the oxygen being consumed and making sure that you're clearing the CO2 and you've not got weird kind of recirculation of gas in the system. That's the kind of only thing I can think of.
I mean, I would say you could do in vivo animal testing, but typically it doesn't seem to be required with these systems because I think there's just too many differences, and so it's taken as a given if you can provide the evidence that you can produce the oxygen concentration you need and deliver it to the patient, that they will get that gas exchange as well as they would on any other ventilator. So I think it's that kind of prior art of ventilation is what covers that application.
Great, thanks so much.
We have time for a few more questions. There's one by Maciej Zajaskowski.
Yes, that's me. Maciej Zajaskowski, yes, that's my question.
Okay, I can read it here in the chat. So after the pandemic is relatively calmed down, how do you plan to efficiently and safely disinfect the respirators for use in non-COVID patients? So as we said before, the exhalation pathway and at the mouth both have filters in it, so they shouldn't need more regular disinfecting than any other device. Those viral filters are very efficient. But we do have a protocol for cleaning the system. It effectively involves pushing a cleaning fluid through the whole system as if it was air. The whole system is watertight as well as being airtight, so you can push a cleaning fluid through, dry the system out again, and then it can be used again. If we needed to, and there may be some requirements regarding transferring from emergency use approval to a more long-term CE marking type approval, that we could just replace, for example, the exhalation pathway. That's about £100 worth of parts at most, and would take about 5 minutes to do. So that's a very easy thing to replace if it was needed.
I have a question actually for Jennifer and Michael, I think would be most suited to answer that question. And I was wondering, so one of the things that I thought was really remarkable is how you managed to do this project in a really short period of time, where others, also very well-educated and capable engineers, struggled with some aspects. And I was wondering whether you could give us a few insights, and you've already addressed this somewhat in the talk, why or how you think that being in a bioengineering department was particularly helpful to make this possible.
So one of the things is we've used a lot of the components inside the system, inside several different systems. So the prototypes that Jakob showed earlier on, a lot of those pieces were actually from different systems we had been building before. And then having that expertise and having that knowledge of having built different systems, we brought in a few of those expertise in, and then we relied on Jakob to bring in his expertise on how ventilators work and what they should do to get that going.
I guess from my perspective is, so, I mean, I was, I guess, classically trained as a mechanical engineer in my bachelor's degree. So I really didn't have much exposure to like the biology and the physiology. And what bioengineering has allowed me to do is really get a good appreciation for that nuanced side of engineering, particularly obviously in bioengineering. And I think that having that interdisciplinary aspect to bioengineering where I can understand, I can speak both languages. So I can speak the language of biology and physiology, but I can also speak the language of engineering. And by melding the two of those together, you really have the skill set needed to be able to do a project like that. So I think really bioengineers are really uniquely suited for a project like this.
Thanks very much. And I think with that, we have to wrap it up because we're already over time. But I think the interest and the compliments and the questions just go to demonstrate that you four and all other people in the team have done a great job. So thank you very much for taking the time off your very busy schedules with very little sleep, as far as I understand, to give this talk. This was very interesting and helpful. Thanks to all the many people who are still in the talk and have joined. I think our next talk is on the 9th. So next week, same time, we have another member of the department, Dr. Firat Buda, talk about the work that his group has been doing on COVID during the pandemic. So thanks very much, everyone. Enjoy the rest of your Thursday, and I hope to see all of you next week.
Thank you.
All right, thank you very much.
Yeah.